The overall goal of this procedure is to measure the effects of stress conditions on translation initiation of mammalian cells and tissues by polysome profiling. This is accomplished by first treating mammalian cancer cells with an anti-cancer drug and then preparing cell extracts. The second step is to make sucrose gradients. Next, the cell extracts are loaded onto the sucrose gradients and ultracentrifuged to separate ribosomal fractions. Ultimately, Western blot analysis using specific antibodies is performed to assess the association of tested proteins with polysomes.
The main advantage of this technique over existing methods like metabolic labeling is that it allows us to assess the effect of stress conditions on the initiation step of translation.
This method can help answer key questions in stress therapeutic field, such as whether and how anti-cancer drugs affect translation initiation.
The implication of this technique extends toward cancer therapy because it helps to reveal new regulatory pathways of translation initiation.
Though this method can provide insight into translation initiation regulation in cell cultures, it can also be applied to other systems such as isolated tissues, including brain and tumors.
To prepare the automated density gradient fractionation system, first wash the tubing system of the apparatus with 0.1% SDS for 5 minutes. Then